Meiosis by William Dwyer, period C. Introduction. Meiosis is the genetic process of creating four genetically diverse cells, with half as many chromosomes as normal cells. These cells are reproductive cells used in sexual reproduction. This ensures that all offspring are genetically unique from one another, allowing for proper adaptation to environments. Interphase. Before meiosis can begin, the cell must first go through interphase. It's identical to interphase in meiosis and has three phases: G1, S, and G2. The G1 phase is for cell growth, with the cell becoming larger, creating organelles, and creating molecules to use later on. Phase S is for duplicating the cell's DNA, and also duplicating the centrioles for cell division. G2 is for additional cell growth, where the cell will prepare proteins and organelles for cell division. Prophase 1. The first step of meiosis is prophase 1. During prophase 1, the nuclear membrane breaks down and the chromosomes condense. Each chromosome is made up of two duplicated sister chromosomes, meaning they are identical to each other. These chromosomes then overlap each other, forming tetrads, or groups of four chromatids. The paired chromosomes then exchange segments with each other, in a process called crossing over. Crossing over can occur up to 25 times per pair. Metaphase 1. The next step in meiosis is metaphase 1. During this phase, tetrads line up at the metaphase plate or the equator of the cell. Spindle fibers, formed by the centrioles, attach to every individual chromosome. This is different from mitosis, in which every individual chromatid would have a spindle fiber attached. The orientation of chromosomes in each tetrad is random, increasing the genetic diversity of the four produced cells. Anaphase 1. The next step of the process is anaphase 1. During anaphase 1, the spindle fibers retract and pull the tetrads apart, separating the chromosomes to both sides. Unlike mitosis, the chromosomes do not separate into chromatids and remain connected to each other by the centromere. By the end of this step, the chromosomes are fully in separate halves of the cell. Telophase 1. After anaphase 1 is telophase 1. During this phase, the nuclear membrane reforms partially around the chromosomes on both sides. Cytokinesis, or the division of cytoplasm, occurs in this step, splitting the cell into two smaller cells. Unlike mitosis, the chromosomes don't decondense in this phase and instead remain condensed. After this step is complete, there are now two separate cells with their own chromosomes and centrioles. Prophase 2. After the four steps of meiosis 1, the four steps of meiosis 2 begin with prophase 2. During prophase 2, the partially reformed nuclear membranes break down again. In both cells, the centrioles move to opposite sides of the cells. Unlike in prophase 1, however, the chromosomes do not pair up and form tetrads, instead staying separate from each other. Metaphase 2. The next step in meiosis is metaphase 2. In this phase, the individual chromosomes line up at the metaphase plate. Spindle fibers from the centrioles attach to every individual chromatid. Like metaphase 1, the orientation of individual chromosomes is random, meaning that any individual chromatid could go to either side of the cell. Anaphase 2. After metaphase 2 is complete, anaphase 2 begins. While meiosis 2 is very similar to meiosis 1 in many ways, anaphase 1 and anaphase 2 share the most similarities. During anaphase 2, the chromosomes are pulled apart by spindle fibers, resulting in individual chromatids pulled to both sides of both cells. Telophase 2. The final phase in meiosis is telophase 2. During telophase 2, the nuclear membrane fully reforms around the chromatids on both sides of the cells, and the chromatids decondense, similar to mitosis. Cytokinesis occurs again, dividing both cells. By the end of telophase 2, there are 4 cells with half as many chromosomes as a cell created by mitosis would have. Conclusion. Meiosis is an essential process in the human body because it ensures genetic diversity. Without it, sexually reproducing species would not be able to adapt to their environment as easily and efficiently.